introduce the biology and the properties of cytokines, to talk about or to try to group the major functional roles of particular cytokines, because there's around 80 of them, so it's impossible to know every single cytokine. To understand what happens when cytokines are released, the response to them, and then to understand that cytokines have very diverse multifunctional actions, and some of them can in fact have opposite effects. They can both stimulate and have negative downregulatory activities. And then just to give you an example of a type of cytokine network interaction. So we first need to understand the general properties of the cytokines. So as I said, there's up to 60 different kinds, and there are more being discovered every year. They, through their release and as ligands and then binding onto receptors, they induce what's called a cascade effect. The cascade effect you already learned with complement, where A activates B, which will activate C, and so on. Many cytokines are pleiotropic, which means that they have multifunctional roles. So a cytokine like TGF-beta can have effects in the brain, it can have effects on bone remodeling, it has an important effect in wound healing, and so on. The secretion of cytokines is limited. So there's a burst of cytokines, and because if the production of the cytokine persists, it can lead to chronic inflammation. And so this is, if you remember 2 lectures ago, when I talked about the stimulation of naive T cells, when they see a dendritic cell, and they form all those connections in terms of the antigen MHC, the co-stimulatory molecules, and the adhesion molecules. What that essentially does is forms a tunnel between the antigen presenting cell and the responding naive T cell. And that allows for a focused direction of cytokines between the, say, the activating dendritic cell and the responding T helper cell, so that the cytokine doesn't diffuse and maybe affect nearby cells. The production of cytokines, when they are made de novo, takes time, 24 to 48 hours. This is because you need transcription, you need an opening of the gene responsible for the cytokine, you need transcription, translation. This is why the adaptive immune response takes longer to operate compared to the innate immune response. And many cytokines can be made by multiple cells. Some of them are made by specific cells. And like I said, they can have different functions depending on their context, whether it's inflammation, whether it's brain development, or whatever, and also depending on the types of other cytokines operating at the time around them. So it's a nightmare to try to understand the role of these things. All I can suggest is that you only understand or learn these particular cytokines in particular functions. In other words, look at the inflammatory cytokines, look at the cytokines involved in cell proliferation, the cytokines involved in winding down the immune response and in wound repair and so on. So as I said, they have multiple functions. Some of them include regulating cell growth, in other words, stimulating proliferation of, for instance, once a naive T cell sees antigen with co-stimulation from a dendritic cell, it will stimulate that naive T cell into an effector cell and also increase or induce that effector cell to proliferate, to increase in numbers, and then also change its telomere length so that it increases its half-life. In other words, it persists in the body for longer to perform its immune functions. Some cytokines, which you'll learn in 2 lectures' time when I talk about TB interactions, will induce gene regulation changes in B cells to induce isotype switching, to cause the B cell to switch from making IgM to IgG, IgE, and so on. Some cytokines directly by binding to receptors can induce those cells to die by apoptosis. This is, have you heard of FAS ligand? Yeah. Some cytokines during the inflammatory response can have changes on the hypothalamus to induce fever, to raise body temperatures. Raising body temperature makes it more hostile environment for microbes circulating in the body to survive. Some cytokines like TNF-alpha cause cachexia. In other words, they cause muscle wasting. This is why patients who have advanced AIDS look like skeletons, because basically the cytokines, the TNF raging through its body is basically eating up all its muscles. And then when you have an inflammatory response, as soon as an inflammatory response is initiated during, say, an infection, then you also have cytokines that start to wind down that inflammatory response. Because if you don't have those negative regulators there, then you get chronic inflammation, which is just as damaging to normal tissues. Historically, the cytokines have been given many different names. First of all, in the early stages, when immunologists didn't really know or understand about these signaling proteins. And what they did was they cultured, say, monocytes from blood, stimulated them with substances like LPS or lectins, and then took supernatants from these cultures and see what they did. For instance, if they added them to other blood cells, they might see that they induce proliferation. They might induce chemotaxis. And they call these factors. And because many of these, over time, they managed to look into these supernatants and isolate various proteins, and then see in more detail what these particular functions of these proteins were. Because many of these substances were extracted from monocyte supernatants, they were called monokines. Then later, during the advent of the HIV epidemic, there was a breakthrough in the discovery of the virus when they managed to isolate a particular cytokine that allowed lymphocytes to grow in culture. And because they could then allow for expansion of lymphocytes, they could then see this HIV virus infecting these lymphocytes. And so then from these development of lymphocyte culture, you could then isolate more substances coming from these lymphocytes. And so they're called lymphokines. And this is the more general term now we use for cytokines, because we understand with the importance of T helper cells that most of these cytokines come from T helper cells. But around that time, people said we have to try to standardize these names. We've got monokines, we've got lymphokines, so let's just give them a general term. We'll call them interleukins. So every 5 years or so, there's an international body that meets to review novel proteins discovered by different labs that have different functions. If they all come to a consensus that everybody's working on the same thing, they give it a name. So they call them interleukins. And so we have interleukin 1, interleukin 2, interleukin 3, and so on. But each interleukin might have another name based on its discovery initially by that lab. So interleukin 2 is also called T cell growth factor, and so on. A third, an important next group of cytokines are called interferons. And these were discovered not by immunologists, but by virologists. Because in the 50s, the way to grow, and they still do this, the way to grow flu virus is to inject 4-day-old chick embryos with the virus and then harvest the amniotic fluid. And this because the virus grows on the amniotic membrane. But when they took this fluid and they added it to cultures of virus-infected cells, they found that this fluid inhibited or interfered with the growth of the virus. And so this generated a lot of excitement because this we now thought that we had the equivalent of an antibiotic for viruses. And so this is why they're called interferons. But subsequently, it wasn't shown to be as a magic bullet like antibiotics. But nevertheless, particularly with this current COVID-19 pandemic, we realized the importance of interferons because many of the fatalities of this virus are due to the inactivation of interferon production by the virus. And then we have the colony stimulating factors, which I think Dr. Rehan might have told you about in hematology. These are GM-CSF, or granulocyte macrophage colony stimulating factor, or GCSF, which induce the proliferation and differentiation of myeloid cells, granulocytes, and so on. But those are also cytokines. In terms of functionality, we have various terms for the activity of cytokines. So one is that many cytokines are pleiotropic, which means that they function in many different kinds of situations, whether it's bone marrow development, neural development, embryonic development, or whatever. All of that can be mediated by the same cytokine. Redundancy refers to the backup system in biology in that no one cytokine has a crucial function. In other words, if you're unable to make that cytokine, its activity can be replaced by 2 or 3 other cytokines. Antagonistic refers to the fact that cytokines might counteract each other. And this is particularly prevalent when I talk about Th1, Th2 cells in the next lecture. Cytokines may be either additive or synergistic. Additive refers to the fact that if, for instance, as you've learned in pathology, that during an inflammatory response, you have IL-1 and TNF-alpha together, then that the effects of both, say, doubles the inflammatory response, right? Synergism, and this is in the case if you have a cytokine like IL-12 and interferon gamma, synergism refers to an exponential additive effect. So you have a super increase in activity. This is due to the fact that the one cytokine might induce more receptor expression for the second cytokine. So the responder cell will then increase its activity much higher due to the activity of the first cytokine. Then cytokines might either have an autocrine action. So in other words, the cell itself has a receptor for that cytokine. So it produces the cytokine, and this can come back and stimulate it to make more cytokines or whatever. Paracrine refers to a normal mixture of cells where production of the cytokine will stimulate nearby cells. And then endocrine is similar to hormones, where the cytokine can circulate through the system and then activate cells in different parts of the body. So now we come to the major functional groups of cytokines. So there are cytokines that are involved in proliferation, what I call pro-inflammatory and inflammatory, and then anti-inflammatory and wound repair, and then cytokines involved in B-cell antibody production and growth. So first of all, in terms of proliferation, the 2 major cytokines we know of are Interleukin-7, which is involved in the stimulation of memory cells and also immature T cells like thymocytes. So during thymic development, IL-7 is what generates all of those thymocytes as they pass through the thymic education. IL-2 or T cell growth factor is an important cytokine for stimulating the proliferation as naive T cells become activated effector cells. So this cytokine induces that proliferative response. Why we separate cytokines as pro-inflammatory and inflammatory? Pro-inflammatory are cytokines which induce the production of inflammatory cytokines. So in other words, when you get the generation of inflammation, you get the production of these cytokines like IL-12, interferon gamma, IL-17, which can then draw in inflammatory cells into the site and then stimulate those inflammatory infiltrates to produce effectors of inflammation, like TNF alpha, IL-1, and so on. But still, many of these pro-inflammatory cytokines can participate during the inflammatory response. So they can also act as inflammatory cytokines too. The anti-inflammatory and wound repair, the two major cytokines of these are interleukin 10 and TGF-beta. So these counteract the effects of the inflammatory cytokines, and they can also prevent their production. And then we have the cytokines which are induce isotype switching from IgM to IgG or IgA, whatever. These are IL-4, 5, and 6. Notice here, though, that IL-6, even though it has an important role in isotype switching, also has an important role in inflammatory response. So this is a reflection of its pleiotropic effect. So back to these interferons. This is the first major group of interferons that were discovered that had effects on interfering with the replication of viruses. And there are two major groups, interferon alpha, of which there are around 7 or 8 different subtypes, and one single member interferon beta. Interferon beta now is an important therapeutic agent for treatment of, well, still experimental, but for treatment of diseases like multiple sclerosis, hepatitis C infections, and so on. Interferon alpha, we now understand that even though this type 1 interferon was essentially more antiviral, we now recognize this type 1 as also having important effects on regulating or stimulating immune responses. For instance, with interferon alpha, it's now been shown that patients with cancer who receive radiation therapy or chemotherapy have better effects if they have interferon in their system. So somehow the interferon complements the anti-cancer treatment. Interferon gamma, which you'll learn about in the next lecture, is an important immunomodulatory cytokine. And then the third group is relatively new. This was discovered about 15, 20 years ago, so we still don't understand much about this group. These are the type 3 interferons, but they are clearly antiviral, and they also show to play an important role in this current COVID-19 case, because we find that patients lacking the ability to make these cytokines are particularly at risk of getting serious COVID-19. So I guess this is a repeat of what pathologists told you with these inflammatory cytokines. First of all, the generation, you've already covered this, but the generation of inflammatory cytokine production stems first from the recognition of antigen, whether it's a microbial PAMP or a damaged cell PAMP, is recognized by the PRRs of these sensor cells, the antigen-presenting cells, right? So in this case, you've got a microbial infection, the PAMPs on so the microbial glycolipids or whatever are recognized by the PRRs on the macrophage or the dendritic cell. And this can either stimulate the inflammasome to create the functional IL-1 and IL-18, or it can stimulate the important transcription factor NF-kappa B and AP1 to produce inflammatory cytokines. Then you can also get a type of opsonization in the case of these complement receptors, C3b and C4b, which tag the microbe and then activate the complement receptors on the macrophage, which can also have similar effects of stimulating NF-kappa B and so on. And similar opsonization effect comes with antibodies, because these macrophages also have receptors, just like they have receptors for the complement components. So this can also stimulate NF-kappa B, right? We've already talked about the inflammasome. And so the three major or four major inflammatory cytokines are interleukin-1, which stimulates many things like adhesion molecules on endothelial cells to allow for extravasation, stimulates macrophages to be more phagocytic, to produce more lysosomal granules so it can degrade phagocytosed particles a lot more efficiently, and so on. TNF-alpha has multiple effects, but again, many effects similar to IL-1, but also, like I said, there are around 12 different kinds of TNF receptors. And many of these receptors can result in either of two pathways. The one pathway is the generation of an inflammatory response through activation of NF-kappa B. The other pathway is the generation of a so-called death pathway, inducing a cell to commit suicide, to undergo apoptosis. So this is a mechanism for this particular cytokine to kill things like tumor cells. This is why it was called tumor necrosis factor, or to kill, say, virus-infected cells. And then again, interleukin-6 has similar activities to TNF-alpha and IL-1, but also interleukin-6 is one, well, the others two, IL-1 and TNF-alpha, but interleukin-6 is what is referred to as an acute phase reaction, that during an ongoing inflammatory response, the detection of IL-6 in the blood is an important marker for an ongoing inflammatory response. This cytokine IL-8 is also an important protein producing an inflammatory response. It's unusual because it's both a cytokine but acts primarily as a chemokine. So another name for it is the chemokine ligand CXCL8, but it's also known as interleukin-8. And its major effect is to be a chemotactic agent for inflammatory infiltrates during an ongoing inflammatory response. This just shows the sequence of events that happens when you have cytokine production. So you have an infection, the PAMPs get recognized by the PRRs on the sensor cell, the antigen presenting cell. These cytokines stimulate the naive T cells to become effectors and then also proliferate and expand in numbers. These stimulated effector cells will then make cytokines which can go back to stimulate the antigen presenting cells themselves, to induce them to upregulate, for instance, class 1 HLA and class 2 HLA. In other words, to increase their antigen presentation, to change their type of cytokine receptors so that they would be directed to a different part of the body where the infection actually took place, and so on. And then they can also produce cytokines which will directly kill the invasive threat. Like I said, once you get the generation of an inflammatory response, you need to have cytokines there to dampen it down, because if you don't, you can start damaging normal tissue. I won't cover it in this lecture, but a big field of research at the moment is looking at a set of proteins called resolvins, which actually resolve the inflammatory activity. But many of these anti-inflammatory factors, particularly IL-10 and TGF-beta, are produced by so-called regulatory T cells. The production of these cytokines prevent cellular immune responses against commensal bacteria, so they promote peripheral tolerance, as well as winding down an inflammatory response that happens. Then you have other factors like these complement negative regulators. Remember from the complement lecture, that prevent the inappropriate activation of the complement cascade, either to break down the C3 convertase complex before it can go into the membrane attack complex, or to prevent the binding of C3 onto a cell to prevent the alternative pathway from forming, and so on. Also, these cytokines, particularly TGF-beta, it promotes fibrotic development, so promoting wound healing after cellular damage as a result of an inflammatory response due to complement and TNF-alpha and so on. And then finally, I just want to talk about the cytokine networks. There are many different kinds, but this is just one example. That first of all, when you get an infection, like say a bacterial infection, this will stimulate the macrophage, which has taken up that bacteria through its PRR and stimulate it to produce this cytokine interleukin 12 and interleukin 15 and these type 1 interferons. Both interleukin 12 and interleukin 15 will stimulate the growth of natural killer cells, and the stimulation of the natural killer cell then induces it to produce interferon gamma, because macrophages themselves don't make interferon gamma. So by stimulating the macrophage with interferon gamma, that induces the macrophage to be more phagocytic and more deadly to phagocytosed material, but also to make more of these NK cell stimulating cytokines. The interferon gamma will then also stimulate the T helper cell into this particular subset called Th1 cells, which you'll learn about the next lecture. And that Th1 cell is predominantly the major interferon gamma producer. This is just a summary of all of these different cytokines involved in the inflammatory response, and that's it. Are there any questions?
No, sir, nothing online.
No. Okay.
Doctor, this lecture
Sorry? No.
There's one question, Dr. Gao. Winston's asking, when the dendritic cells release cytokines to stimulate naive T cells to become effector cells, does it cause both cytotoxic and helper T cells to become activated and migrate? Or does it depend on the MHC the antigen was presented on?
So the question is, when a dendritic cell stimulates a naive T cell to be an effector T cell,
This one.
released to stimulate naive to become effector, does it cause both cytotoxic and to become? Yes, it does. So when the naive T cell is activated, so that's dependent on whether the antigen is presented on class 1 or class 2, obviously, right? So if it's class 1, it'll stimulate the development of a cytotoxic T cell. If it's class 2, development of T helper cell. This can all happen by the same dendritic cell because the dendritic cell can present both class 1 and class 2 antigens. So once these two cells, the cytotoxic T cell and the T helper cell becomes effector cells, they can then go off, proliferate, and then perform their functions.
All right, thank you so much.
All right. So I won't see you before your midterm. Good luck with the